Good day. My name is Rafali Vashev. I am the Vice President of Innovation and Technology at DNA Genotech, a subsidiary of OraSure Technologies. Together, we offer molecular solutions for collection and stabilization of biological samples for transport to laboratory settings. Our parent company, OraSure Technologies, offers point-of-care infectious disease diagnostics. Our molecular solutions devices were developed to solve market challenges and have been used around the world under difficult conditions to conduct population-based genetic studies. The devices have also been used both clinically and in research, focusing on human and microbiome applications. Our focus is to ensure a quality sample arrives at the lab for accurate testing. Based on our years of experience, we were well positioned to support the pandemic effort. We have been busy generating data and supporting our customers to demonstrate that our proven technologies are well positioned for these use cases, from sample collection to diagnostics. Our molecular collection devices, which come in different form factors, the OraCollect RNA, a sponge-based device, and the Omgene Oral, a saliva-based device. Each of these devices is non-invasive and easy to use. The stabilization chemistry allows for the recovery of high-quality DNA and RNA, even after ambient temperature transfer. One key study that we completed earlier this year was to demonstrate that the guanidine-free consumer-safe chemistries we use in our products can inactivate the SARS-CoV-2 virus immediately upon collection, allowing for safer transport to the laboratory and an additional precaution once in the laboratory setting. In this slide, you can see the positive control, the black bar, and then when the virus is added to Oragene, OraCollect DNA, Omnigene Oral, OraCollect RNA, or Omnigene Gut, the level of virus detection is at baseline, meaning that there is no detectable virus. We have developed a novel chemistry that works with our stabilization products that allows the bypass of traditional extraction. PrepIt Q2A is a simple liquid-based protocol that can be used to process samples in high throughput. The processed samples can be then used in downstream applications. Our stabilization chemistries are also compatible with many traditional extraction kits. Here, I share a few selected examples of kits being used today for SARS-CoV-2 work, which we have demonstrated work well with our collection devices. The pandemic has highlighted the intersection of host and pathogen genetics. Our suite of products is ready to be deployed for the study of both. It is now my pleasure to introduce Professor Dr. Andy Goren, with whom we had the privilege on working on such a human genetics project this year. Dr. Goren has over 20 years of experience in research, development, and commercialization of breakthrough therapies and diagnostics. Among the list of firsts in dermatology, Dr. Goren was the first to develop a clinical genetic test for predicting androgenetic alopecia. Dr. Goren is actively involved in the development and commercialization of several drugs and medical devices, including new therapies for female pattern hair loss, chemotherapy-induced alopecia, and excessive hair shedding. His latest research involves the use of anti-androgen therapy for the treatment of COVID-19. Today, Dr. Goren will share with you details of his latest research in the presentation titled COVID-19, Androgens and Androgen Receptor Genetics. Thank you.
Hello, and welcome to this webinar. My name is Dr. Andy Goren, and I wanted to share with you some of the research that we have lately worked on COVID-19, androgens, and the androgen receptor genetics. This is a very interesting evolution of actually how dermatology meets genetics and how actually the human or the host genome is involved in pathogens and this pathogen-host interaction. So COVID-19 obviously has been on everybody's mind, and it's been devastating globally impact. And I think that some of the interesting insight have come actually from genetics, both the human genetics as well as the pathogen genetics. I don't have any conflict to declare. I do have quite a bit of affiliation collaboration, that quite a few of them, and have helped make this project come to fruition. DNA Genotech has contributed quite a bit, both on their devices and expertise. I'm affiliated with Applied Biology, and I work with various universities around the world and companies, primarily in dermatology, but in the last probably 6 to 9 months, it's been shifted into COVID, and interestingly enough, there's a cross between dermatology and COVID. So for me, this journey into COVID really started with the quarantine, and interestingly enough, I don't know if a lot of people know, but the word quarantine comes from Venetian during the plague, where it means really 40 days. It was 40 days required to be quarantined when you arrived on a ship, just because of the plague. So to me, that's where the whole journey started, being locked like everybody else in the quarantine and thinking about COVID. And one of the things that struck me is looking at sort of the outliers. And in the outliers, what you see is early on, it was observed that men are disproportionately affected compared to women, both in mortality rate and disease severity. ICU admission primarily. And prepubescent children were almost not affected at all, which was also very interesting. Some of the science behind it was fairly early on discovered, because there's experience with SARS-CoV-1, or just SARS. And SARS-CoV-2 primarily affected type 2 pneumocytes in the lungs. There are other mechanisms, both neural and non-neural even. When I mean by neural not learned, so one of the symptoms is anosmia, where you cannot smell. That symptom has come from actually ability of SARS-CoV-2 to enter into neural sites. So there's some controversies and there's some papers around it, but primarily it's type 2 pneumocytes in the lungs. What we do know is SARS-CoV-2 requires the ACE2 receptor on cells on type 2 pneumocytes to express, as well as TMPRSS2 to cleave the S-spike protein. There are other ways, probably, furin protease possibly, to enter the cells. And we know that because if we actually knock down in cells in animal models, where we have cells that have all sorts of inhibitors, we still have some SARS-CoV-2 infectivity, albeit much lower when we block ACE2 and TMPRSS2. So TMPRSS2 is probably the most implicated enzyme to prime the SARS-CoV-2 S-spike protein to enter into cells. And one of the things that we know, and this relates directly to human genetics, is that the only known, and again, I'll emphasize known, promoter is the androgen response element. So that's very interesting to us, because we worked on androgen for a long time, and this is, in a second, I'll show how this connects with hair and alopecia. But ACE2 is the other receptor that's also X chromosome linked. So again, both of them are in the X chromosome. What's interesting is because most of the work we've done really is of hair research and androgens in male and women, that's our dermatology background, it immediately let us think, oh, androgens. Well, androgens are implicated in hair loss. And so in men and in women, more in men with male pattern baldness, which is on the X chromosome, the androgen receptor. There are some mutations in it that are actually fairly predictive of the phenotype of the male pattern baldness. With females, a little bit less so. It's a more complicated phenotype, but in both cases, it lets us to realize androgens are involved. Can we find some correlation in the population that are in the ICU? So we, as far as we know, we're the first one who actually published back in March of this year. What does androgenetic alopecia have to do with COVID-19? So we had this theory that we will find more patients, both male and women at the time, that are in intensive care or that are deceased because they have heightened androgen sensitivity. So that was really an interesting start for us for this journey. It seems like a long time ago because this is 6 months, but in COVID time, it's a really long time. And since then, we have authored over 20 manuscripts. I think 15 or 16 have been published already. So it's been kind of a record set time this year. As far as studies, so we started the first observational study was done in late March, early April. We had a small group of patients that we looked at that were in the ICU. It was a really a small observation, but when we noticed the disproportionately males that were in the ICU had severe androgenetic alopecia. And what's important about it is you'll say, well, as men age, a lot of them do have androgenetic alopecia, true, but the mean age was 58 years old and 71% of them had androgenetic alopecia. Whether would you expect in that population, 31 to 53%. So that was really interesting, and it was kind of the first clue that we had that, wow, maybe we are right about this androgens. So we obviously expanded these observational studies. We've gone to a multi-center observation. We had 175 subjects at this time, and this seemed to hold again, the idea that there's a disproportionate amount of males for their age compared to age-matched controls in the same population or ethnic variation that are presenting bald. Interestingly enough, we published this with several other papers, but we had this eponym that Goren signed, and really the Goren, Frank Goren was the first American physician that we know of that died in the line of duty from COVID, and so we thought this was something to name him after as a sign that both when a patient presents with that baldness, we should be more careful and observe them for higher likelihood to get into ICU, as well as when we design clinical study, this will be a patient population we might want to recruit. There were several other papers because we had some questions that came along the way about, well, whether all men, as I said before, as they get older, there's higher prevalence, obviously, of male pattern baldness, but it was really disproportional when we broke it down by disease severity and hospitalization for the age group, and it kept holding in different populations. We thus kind of concluded the androgen sensitivity might have something to do with ICU in men, but we were really inquiring about women as well, and we have not found that relationship, at least not in the female pattern hair loss phenotype. And the reason can be multiple. Androgen sensitivity in women doesn't necessarily present as female pattern hair loss. So it could be PCOS, can be hirsutism, could be acne, can be whole sorts of different things that can happen. And they're not always observable. It's sort of like the prostate also is implicated in androgen sensitivity, but when a patient presents from COVID-19, we don't necessarily know if they have an enlarged prostate. So it falls into the same category. So we don't know yet conclusively whether women are androgen sensitive. Androgen sensitive women will predispose to higher disease burden in COVID-19. Another observation that we made that related to that early on, and this was a topic that was just sort of evolving, is the racial differences. And there's obviously racial disparity in the standard of care, as well as access to medications and treatment and awareness around the world in different populations. But one thing that did strike us is if you look, for example, at African-American men, they do tend to have more disease burden when it comes to prostate, more disease, more androgenetic alopecia, and a lot of it is actually related to the fact that, as we know, the androgen receptor, they're more sensitive. In fact, they have this CAG repeat in the first axon of the androgen receptor that predisposes them to higher prostate disease burden, as well as, or really benign prostatic hyperplasia, as well as prostate cancer, as well as higher incidence of baldness. So that was not to take away from the fact that there's racial inequality and disparity in care, but the issue was really that we should recognize, and we know that very well from genetic, there's ethnic variations and they can contribute to disease burden as well. So we published that also sometimes in April. Interestingly, there was an Italian study that came afterwards. I can't remember where, I think it was around end of April or May, and it showed that men who are prostate cancer patients on androgen deprivation therapy seem to have much less likelihood of appearing into the hospital for COVID, as well as the disease burden seemed to be a little lower. It was, again, an observational study. So we started actually conducting our first study, which was a prospective diagnostic study to predict COVID-19 disease severity in hospitalized patients. And that was a genetic study based on the androgen receptor. We looked at the CAG repeat in the first axon, which is generally predictive of androgen sensitivity. So it not always predicts the androgen phenotype. So for example, we will not be able to predict with that whether a man is likely to go bald, but we could predict with that whether a man will respond to androgen therapy fairly well. So we looked at that, and and one of the things that we recognized when we developed the study, this was not a normal anymore study of, hey, let's just collect some DNA from patients and see what happens. This study, actually, we're looking at infectious samples. This was a saliva collection. We had to use a collection device that both was CE and FDA cleared. Unlike some of the other swabs and some of the collection devices that you can use, they're sort of class 1 devices. They really are not meant for infectious diseases. They also require some development of both collection protocols, storage protocols, shipping protocols, and processing protocols for all this, or data analysis, that are for infectious diseases. And I think one of the things that we learned from this is post-COVID-19, I think a lot of the DNA samples that we're going to collect for different studies are going to be treated as infectious diseases, whether they are or not, because we just don't know. I do think that would be an important point. So we used the CAG marker, again, that I mentioned. This was in men, so there's only one X chromosome, unlike in women, which is more complicated genetics, but this was focused again in men because that's where we saw the first association. And we used next generation sequencing to do that. So I'm going to present the data from the first study that was actually published, but we are now conducting it as part of a multi-center study in US, Spain, and Brazil. What we found is really a striking difference between the median hospital stay and the percent admitted to ICU. So in men with a short CAG repeat, and 22 was actually, it's the mean CAG or average actually CAG in that population. So that's the cutoff that we chose. So men with the short CAG had an average or median hospital stay of 25 days compared to 48 days with men with the long CAG. Similarly, when you look at the ICU admission rate, the short CAG was 14 days and the long CAG was 24 days. So that's pretty striking, and it was statistically significant even though it was 60-something patients. So that was really encouraging because that was the first direct proof we have that there is actually involvement between androgen receptor and COVID-19. But there is a question that somebody probably is already thinking on their mind. Well, usually we think that short CAG is what predisposes you to androgen sensitivity. And so we were a little bit confused. What's going on here? Why is people who are less androgen sensitive this has happened to? And initially we were thinking, well, okay, well, we can dismiss that because it doesn't really matter. Maybe it's behaves different in different tissue in the scalp and the prostate. But what we recognize actually that, yes, that is true. And androgen sensitivity is tissue dependent. So in the skeletal muscle, actually, the increased CAG repeat length confers higher activity. However, what we did learn here is that the AR expression, and I think most geneticists know that, they're dependent both on co-regulator and co-activators. And it happens to be that in the lung, PRMT6 is highly expressed co-activator of the androgen receptor, especially in the pneumocytes, we found that. So with that, the PRMT6 co-activator actually interacts in the N-terminal domain with the CAG repeat lengths. And the longer repeat actually expresses more androgen receptor. So that was very interesting or more active androgen receptor we have. So that was really interesting because now we have at least a theory what happens in the lung and why is it different than what we see in the prostate or in the scalp. It's just different, and so is the muscle. So we have at least a theory why what we're seeing is possible. Continuing on the androgen theory, we're actually conducting some prospective studies with 5 alpha-reductase inhibitors. We primarily chose those just because we have experience with them in dermatology, but also the PRMT6 is ligand dependent. So we would know that it's not just, unlike the castrate-resistant prostate cancer, which sometimes does not read actually the ligand, PRMT6 does require the ligand. So when you have the longer CAG predisposes you to higher disease severity, it does require DHT. So targeting DHT would seem the obvious choice. There's also drugs that have been for a long time approved and fairly safe, Dutasteride being one of them. So in the first study that we have done while we were recruiting patients, this was first an observational study and then became also a prospective randomized control study. We had 403 patients that are screened. Of them, 113 had androgenetic alopecia, 48 were using 5-ARI, and 65 were not using 5-ARI. We used propensity score matching with a greedy algorithm of and a 1-to-1 ratio. And amazingly, 20 out of 29 of the baseline parameter, or the, sorry, 20 out of 29 of the outcomes of disease severity were significantly reduced in the group that are using 5-ARI. So this is a paper that we submitted recently, but it's extremely exciting to see the characteristic of this population, whether it's fever, low-grade fever, anosmia, all these things have been extremely low in the group that's on 5-ARI. So this is the first direct evidence we have that actually the Dutasteride used long-term could be a prophylactic for COVID-19. So just some other areas that are interesting potentially and also related to androgen receptors. So once we talked about androgen receptor, it occurred to us there's actually day length effect, the clock genes, androgen and androgen expression. So we actually put the paper on this we published recently that could potentially, or it's a question, or we're predicting whether the clock genes, which depend actually on the day length, and this has nothing to do necessarily with summer and heat and all that, it's just the length of the day changes, androgen expression actually changes. And there's even seasonal variation in PSA. We know there's seasonal variations in the animal kingdom. So this was very interesting to us because as we're approaching now October, November, day length actually shrinks, and there is actually increased expression of some of these things, which could actually predispose to higher disease severity. Another area of interest that we're looking at is ACE2 and TMPRSS2 in hair. What will that do in the hair, and what potential treatments maybe for hair loss this can uncover. So just in conclusion, COVID really had a significant impact on everyone's lives, and the global crisis is sort of like a war. It accelerated research in so many different aspects of things, and not just for COVID, but also for many other respiratory illnesses. So for us, this was a very intense and obviously sad overall time, but it was also exciting to discover some new things. And one of the things that we did learn that we think is very important going forward will be to really think about, when we design experiments in genetics, is how do we collect samples? How do we ship them? How do we treat them? Because not everything is going forward. Even if you do a commercial DNA test for testing anything that's not even medical, the samples need to be thought of as infectious disease sample. So this is just some thoughts and some ideas going forward, and wish everybody to be safe. And if you have any questions, feel free to contact me. Thank you again for inviting me.